Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base Biotech podcast, a podcast about, well, you guessed it, biotech. And the episode this week is going out on March the 28th. If you are in the UK, it is Mother's Day this weekend, and the clocks also spring forward. So I guess it's not actually Mother's Day, it's Mother's 23 hours. Thanks so much to all the people that listened to the first podcast last week. The response actually was quite excellent and a bit overwhelming, so thank you to both the listeners. No, actually, I'm just kidding. We were in the several hundreds, so a great start and a good base, you could say. The interview requests keep coming in, so that's good as well. I do get lots of requests to do interviews on subjects that I don't cover. One this week was to see if I wanted to interview someone about the cheapest places in Turkey to buy beer. Maybe if I ever start a turkishbeer.com podcast, who knows. A reminder that you can find this podcast alongside a short article on one of our websites, deeptechdigest.com, and you can also get in touch there by emailing me, or you can just do it directly, jim@deeptechdigest.com. And you can also subscribe on Spotify, Apple, Google, and most podcast platforms, and it's on YouTube. Today, again, we have two interviews with Dr. Hernan Bazan on South Rampart Pharma's quest to provide new pain treatment alternatives in the face of the opioid crisis. But first, we're talking about epiproteomics, among other things. I could explain it badly, but it would be far more convincing coming from an expert, and that expert is Ronel Wexler, the co-founder and CEO of Promise Bio, whose AI-driven system combines multiomics data analysis with proprietary machine learning algorithms to identify unique biomarkers and disease pathways, and that enables early precise diagnosis and personalized treatment strategies for autoimmune conditions. The company recently secured $8.3 million in seed investment with backing from Pfizer and AstraZeneca, and the company has been recognized as one of the most disruptive scientific startups of 2024. So I wonder if first you could tell me a bit about Promise Bio and its emergence from stealth.
Yeah, sure. So at Promise Bio, we develop precision medicine solutions for complex chronic diseases, starting with immune-mediated diseases. We do that using our technology that is focused on broad epiproteomics. So in a nutshell, we develop tools for treatment selections. And we do that by looking at the most dynamic and complex biological data, which is epiproteomics, and I'll elaborate on that in a second. A bit about the company. We did our seed round in 2023. Part of our seed round was joined by AstraZeneca and Pfizer that joined as early strategic investors. Our technology was developed by my two brilliant co-founders during years of research at the Weizmann Institute in Israel. I mentioned my two co-founders. Maybe it's a good time to explicitly mention them. So the company was founded based on the research that was performed at the Weizmann Institute by Professor Rifat Merber. She's our scientific co-founder. She's a professor at the Weizmann Institute at the Systems Immunology Department. And Dr. Asaf Kassen, our co-founder and CTO, who developed the technology during his years of doctoral studies at Ifat's lab. And what's unique there is that the lab, and specifically the background of Rifat and Asaf, is very multidisciplinary. So it's not, the traditional biology background. They both come from computer science. Asaf did his bachelor's in computer engineering. And then Ifat's lab is very unique in the sense that they combine expertise in chemistry, biology, and then computational methods. And then this really intersection is what's needed today to solve these hard problems. So they developed this amazing technology, then we got connected and established Promise Bio. So shout out to Asaf and Ifat. We have a great team, more than 50% PhDs from leading research institutes. I would say that it's some of the rationale of how we established Promise and where we decided to take it was also based on my previous experience. So I'm a physician scientist by training, and I started as an electrical engineer, did algorithm development for years. So when I practiced medicine for several years, I met these patients with immune-mediated diseases and saw how the way we manage these diseases is like trial and error. And that's really inconceivable in 2025 or 2024 or 2020. So that's something that I really wanted to change. And then the previous company I worked for called C2I Genomics that developed a blood test for cancer recurrence monitoring, and that was acquired, really broadened the horizons of how precision medicine can drive like real impact based on modern data science and algorithms and machine learning. So combining, these there's a urgent clinical unmet need. There's the right technology to solve these challenges now, and we have the right team. So that's really exciting. We are working with pharma companies already to help them solve some of the hardest challenges around precision medicine. And I'll touch about these kind of specific challenges maybe a bit later. But the idea is that in 2025, the ability to understand biology better dictates that we harness the right tools and use the right data. So it's a data problem, like in many other industries. And we've been advancing in the computational resources and machine learning methods that we can now apply to very complex data and large scale data. And that was not possible before. And this is the advancements that we are now harnessing. So it comes from different perspectives. So sure, there's, cloud computing and machine learning now, but also, again, based on this research that my co-founder has done, we are now able to kind of combine this interdisciplinary deep knowledge and understanding of biology, chemistry, specifically around proteomics, to understand how proteins change after they're created.
And you already kind of gave a passing mention of epiproteomics. I wonder if you could go into a bit more detail on what that is.
Of course. So maybe we should start by addressing what is proteomics before epiproteomics. So proteomics is the field of studying proteins, and that usually stops at the level of protein abundance, meaning how much from each protein is within a sample. But that's not where the biology stops. So proteins, after they're created, can undergo changes. These changes are called post-translational modifications, or PTMs in short. And these modifications, or PTMs, are known to play a key role in biology in dictating what will happen to protein, where and when. And we know in the science world, we've been studying the importance of PTMs for decades. And we know that PTMs are super important. And you can look at it like micro switches, small molecules that are attached to proteins and change and dictate the fate of the protein. So, for example, many people are familiar with phosphorylation, which is the addition of phosphor to a protein, and that sometimes leads to an activation of the protein. On the other hand, sometimes people are familiar with ubiquitination, which is a natural mechanism that sends proteins to degradation. So really these like small changes, small molecules, PTMs, change the functionality of the protein all the way from activation to degradation and everything in between. So providing the additional layer of regulation in our body. So when we look at the collective of modified proteins and unmodified proteins, that's a group that we now can call epiproteome, because now we can look at this both, again, modified and unmodified proteins and study the importance and contribution of these different functional states to disease phenotype, response to treatments, and so on.
And so what's the importance of PTMs when it comes to investigating diseases?
Maybe I'll start with like 3 brief examples. And we've already known that for decades. So 3 examples for how PTMs are known to be important in real world clinical work. So the first one is, and I'm sure you know the term, is glycated hemoglobin, or as everyone knows it, HbA1c, which is the best biomarker for glucose level stability for diabetic patients. So for patients with diabetes, they are constantly monitoring their glucose levels, right? So they do it with their prick test, but also when physicians want to understand whether let's say for an average of 3 months, what was the glucose level like, they are looking at HbA1c. That's a very reliable measurement of the diabetic control. So that's on diabetes. In rheumatoid arthritis, we have a very robust and important biomarker called ACPA, anti-citrullinated protein antibodies, which are antibodies that our body creates against citrullinated protein. Citrullination is another form post-translational modifications. So we know now that these modifications are contributing to the pathophysiological process of autoimmune diseases. And ACPA, or anti-CCP, as it's sometimes called, is a very important biomarker and affects decision-making in rheumatoid arthritis. And finally, just recently, and by the way, diabetes the HbA1c was discovered in the 70s. ACPA is using for the past 20-something years. And recently in 2024, p-tau, or phosphorylated tau protein, was added as a biomarker for early diagnosis of Alzheimer's disease. So for the first time in 2024, they added a new blood-based biomarker to be part of every diagnosis of Alzheimer's. That biomarker is actually a modified proteins. So just, 3 examples from real-world clinical usage kind of perspective. But then the important part is that the way that scientists has been revealing these PTMs was very much based on anecdotal research. So they had 1 modification in mind, 2 modifications in mind, 3 modifications in mind, and they had to go through a very laborious task of studying these on a very narrow basis. So then we asked ourselves, okay, so what if we could now, instead of studying just 1 or 2 or 3 PTMs, we could look at broad range of PTMs. So maybe it's a good time to emphasize that there are hundreds of potential PTMs. So maybe a step back, proteins are built from amino acids, and almost all amino acids can undergo this type of modification. So there's now a huge combination of amino acids and modifications that really adds an exponentially larger amount of data or complexity to the biological data. Again, going back to our notion of it's basically a data problem now. What if we could look at this complex, vast data using our modern tools? So that's what we do. So our basic technology now allows us to look at more than 60 PTMs simultaneously, while conventional science was still focused on between 1 to 3. We are already at 64 now, more than 64. And we do that based on standard data with no extra steps in the lab.
When it comes to precision medicine, what are the current challenges in that field?
Yeah, so precision medicine is, is the holy grail. We all want to be able to give the right patient the right treatment at the right time. Unfortunately, it's not the case, especially in complex chronic diseases. And, we have been making huge advancements in cancer, right? So all cancer patients are now sending their tumor biopsies to DNA sequencing, where if there's a mutation and it's known to be related to a known treatment, a personalized treatment can be tailored. Outside of oncology, that's not the case. So especially in diseases like Crohn's, colitis, rheumatoid arthritis, these are diseases that affect millions of patients across the globe. They receive very expensive drugs. So that's a huge problem, not only for the patients, but only to payers all around the world. We don't have any tools to identify from the ever-growing number of options of expensive treatments. So the biological treatments cost tens of thousands of dollars per year. We don't have the right tools. So that's where we are aiming for. So we are developing tests to allow physicians down the road to get the right treatment for the right patients. When we look at precision medicine, it has several kind of aspects to it. One is the clinical diagnostics world, which I just mentioned, but also if you look at it from a pharma perspective, there's so many questions for the precision medicine groups within pharma organizations. They just don't have any tools that will help them to solve. So for example, if they have a constant challenge to understand mechanistic biology better, and that drives their decision making from the beginning of the drug R&D all the way to clinical trials. So deciding what indications to pursue, deciding some questions around the optimal dosing, and of course, finally, to optimize clinical trial enrollment. These are all different decision points that if pharma companies would be able to stratify subgroups in the population, they will significantly increase their chances of success. So how do we do that? The common denominator is better mechanistic understanding. Better mechanistic understanding and more granular insights dictates looking at the most relevant biological data, the most complex, the most dynamic, and so on, because these diseases are really heterogeneous and dynamic by nature. And then we go back again to, okay, so this is a data problem. What data should we look at? And it boils down to, let's look at the epiproteome.
Maybe we could talk now about your own platform and how that works and where the data come from.
Sure. So our platform has kind of a three-step process. The first part of the process is where we capture, meaning that we get the data. So we work on data that is produced by machines called mass spectrometers. So our first part is that we use our proprietary platform. It's called Prospect. It was published in Nature Biotech in 2023. And that platform allows us to take the standard mass spec data, digital data, and extract more than 60 PTMs simultaneously without the need of any additional chemical enrichment process or any extra steps in the lab. It also means, by the way, that we can reanalyze any previously acquired data. That's really appealing for our pharma partners, but that also helps us because we can leverage any previously acquired data that is in the public domain. So that's the first step. We get mass spec data in and we get dozens of PTMs on a very high resolution in a very robust manner, very scalable, all cloud-based from this digital output. The second step is what we call augmentation, where we are using our proprietary data lake called Epitome, which is kind of harmonizing data from different diseases, different instruments, different tissue types. So we are using our previous learnings from all these analyses, from our academic collaborations, from proprietary data collections that we're doing, from publicly available data, and we use these learning and identify patterns that can now enrich in silico, can do an in silico enrichment of the data that we are analyzing in step one. And then the third step is our reasoning step. So we take the data from step one, we enrich it or augment it with all our previous learnings, and then we combine these two with our set of analytical steps to get biologically relevant and actionable insights. So that looks like, for example, finding molecular fingerprints for response prediction. That could look like finding biomarkers for different disease subtypes or different population subtypes. And again, there are probably 5 more use cases in which we are helping with. So that's a glimpse of how we work. You asked about the sources of the data. So we have 3 sources of data. One is the academic collaborations. So we are working with leading research institutes all around the world to help and solve these challenges that are really coming from the clinics. Second source is publicly available data. There is a growing number of data that is available out there. I wish it was more like genomics data. It's not the case yet, but it's a growing in a number of sources. And then the third source is our proprietary collection of data. We have several multi-center prospective clinical studies, observational, of course, where we follow up on patients with immune-mediated diseases. So we get a very deep annotated clinical data sets with biological samples in multiple time points.
And so how does what you do benefit, I guess, first your partners and then ultimately patients?
Yeah, so pharma companies have these challenges around, let's say, 3 major areas. One is the ability to identify predictive biomarkers. A second one is question regarding to indication selection, expansion, and so on. And a third very common challenge is around dosing. How do they identify the optimal dose? How do they identify what their drug is, or how the drug is affecting the different biological pathways? How does that change according to with different doses and so on? And these are hard questions, especially in diseases where you don't necessarily have, or they don't necessarily have tissues for. They can't, like biopsies from cancer. So in immune-mediated diseases, they usually have some sort of liquid biopsy, whether it's blood products or urine in relevant cases. And to meet these challenges, they, as I mentioned before, they need to go and improve their mechanistic understanding and MOA analytics. And the way to do that goes back to let's analyze the right data using the right tools. So this is how we partner with pharma companies. They come with a question or a set of questions. They come with their data, and we go through our 3-step process that I described and answer their questions. And then eventually down the road, as we kind of grow our data lake from the academic collaboration or proprietary data collection and publicly available data, that would allow us to use our constantly improving algorithms to also find the biomarkers that would then be used to develop our own proprietary products, which will be the treatment selection tests.
What diseases and conditions are you looking at? I mean, does it have multiple applications?
Yes, so it's really a platform play. The ability, if we look at where could understanding proteins and modified proteins could help, the answer is all over. We made a strategic decision to focus on complex chronic diseases. So we start with immune-mediated diseases like inflammatory bowel diseases, rheumatoid arthritis, lupus, and lupus nephritis. We've also been approached with requests to analyze data in chronic diseases that are not necessarily inflammatory in nature, like chronic kidney diseases, other metabolic disorders. There's a huge gap there in our understandings, so we help there as well.
So I guess what are the next steps for the company? Where do you go from here?
Yeah, so we are in a super interesting time in the company's life. So we announced our emergence from stealth. We now have a bunch of projects that are in the oven. So both from our academic collaborations, our own proprietary data collection and our pharma collaborations, we already see super exciting results. I hope that in the next few months, we'll be able to share more publicly what we see. And that would lead us to expanding the company, expanding the partnerships, running bigger trials, and getting closer to commercially available development of products, and also expanding our business offerings.
So now if someone asks about epiproteomics in a quiz, you've got it covered. I always get movie or TV questions when I want music, sports, geography or science. Anyway, a very interesting subject, and you know it's good if Pfizer and AstraZeneca are involved. Now over to the second interview, and it's about providing pain relief alternatives to alleviate some of the problems that have been created by what has been termed the opioid crisis. Dr. Hernan Bazan is a qualified surgeon, and he's also the CEO and co-founder of South Rampart Pharma. And in our conversation, we address the crisis and how South Rampart Pharma is tackling the issues head on. Normally, when I do interviews, I ask about the company first, but before we do that, I thought it might be interesting to look at what you've done so far, because it's quite interesting. So I wonder if you could go through some of the highlights of your career.
Sure. Thanks, Jim. Nice to meet you. And I hope maybe one day we can meet in person. I'm a practicing physician, vascular surgeon in the city of New Orleans at a place called the Opioid Clinic. And I also run our fellowship. I spent a couple of years at the NIH before I went to medical school at Georgetown. And there I had done some research, and there I did quite a bit of research. I did my post-medical school training in New York at Mount Sinai, and then my fellowship in New Haven at Yale. And then in practice, all of these things sort of culminated. As you know, in the late 90s and early 2000s, the opioid epidemic you've heard about in the US is really out of control. And obviously have seen many patients that have had that problem, especially as a surgeon. My father is the head of the neuroscience center at the medical school at the LSU School of Medicine. And they have a big focus on neurochemistry and different CNS disorders like stroke and TBI. And they also had an interest in pain. And so we put that together, and actually with a medicinal chemist in Spain, we did a library of compounds specifically to keep the pain, but to take away the pain relieving properties, but to take away the liver toxicity of what's currently available. For example, with Panadol, acetaminophen, or Tylenol, depending on which part of the world market. And by not being an NSAID, non-steroidal, working to take away the kidney and GI toxicity, particularly the bleeding risk as patients are older. And the whole approach was to make this a novel non-opioid. And so that's what we've set out to do. And so what we've done is essentially characterized how it works in the brain. We know it crosses the blood-brain barrier. We know what part of the brain it works in. It's called the PAG region, which is in the midbrain, which is the pain nucleus, the pain-sensing nucleus, the periductal gray area. And the mediator is something called the AM404, which is actually a clinically validated mechanism of action. So a lot of this is what we published in May of 2024 in Nature Portfolio Scientific Reports describing these mechanisms. And then we also, fast forward several years, opened an IND with the FDA and brought it into the clinic, and just completed a year ago the phase 1 trial, actually with a UK-based company called Quotient Sciences. They're excellent at clin pharm studies. So we were able to do very good, not just safety and tolerability, but also very robust, established the pharmacokinetics, the effect of food, and so on and so forth. And the phase 1 data was extremely encouraging. No serious adverse effects. All the lab values are normal, and it revealed robust PK, very high absorption initially, very high Cmax with a bimodal distribution, a very long half-life. And that's also described in the Nature Scientific Reports paper, the phase 1 data. And so now with the US FDA, we've opened a few different pipelines, and the first one is acute pain. And that's what we're aiming to execute by the end of the second quarter of 2025. And the reason that we're focusing on acute pain is because, as you could imagine, all of these things are happening in parallel. A lot of the science that we're generating, the clinical evidence, and then the capital raise to fund the phase two, but also talking to strategics. And the feedback that we've gotten from them is they really like how well characterized SRP-001 is, and they understand the mechanisms of action. They're very encouraged by that, particularly how it works centrally. And we can touch on that, how that's different than something else that's happening now with the good work that Vertex Pharmaceutical has done with a peripherally acting sodium inhibitor, their NaV1.8 inhibitor, and the emphasis now on generating the human efficacy data. So that's in a nutshell everything that's going on.
What's the history of the company then? How did the company come about?
The company was founded by the scientific co-founder, my father, the head of Neuroscience Center, and myself. And we took the technology, as I mentioned, the library of compounds between the universities, and we have the ability to develop and commercialize the technology. And so we've done all the characterization, all of the understandings of how it gets metabolized, how it works in the brain, the molecular mechanisms, particularly, again, AM404 and how it gets taken up in the brain and how it works in the brain to induce analgesia, pain relief, established the animal models. Along the process, we've been able to receive 2 NIH grants, U.S. National Institutes of Health grants for a lot of this work, which I think is important to recognize because of the strong support that they continue to be. All of this has fueled the preclinical and then opening the IND with the FDA and getting into the clinical development.
In terms of opioid addiction, how big is that problem, and what are the biggest issues in tackling that problem?
I mean, it's a tremendously big problem because it's very variable. There are some humans that take just 1-2 opioid pills, oxycodons, and they're fine. And for when they have a surgery or some trauma or something else, or it's prescribed incorrectly by a physician, that's not really happening that much anymore, luckily, because there's been so much stigma against it and so many pressures against it. And then there are people that take a few narcotics and then unfortunately their life changes. They have something in them that make them become addicted, and then it leads to opioid use disorder, OUD, and different types of abuse patterns. And so the issue in the pain space is that opioids work up here. They're very good at relieving pain, whereas what we have currently available are down here, NSAIDs and Panadol, Tylenol. And then there's a space in between. And this is where there's a huge gap. And so, for example, now the great thing is we have a precedent. So Vertex Pharmaceuticals in Boston has done a very good job of pushing forward a peripherally acting NaV1.8, sodium voltage gated 1.8 inhibitor. And the reason that I say that this is actually very important for the pain space is because what they demonstrated is how the market views this. So when they were approved in late January for acute pain, the market cap for Vertex changed 8 billion in one day. And that was really important because up until recently, the last several years, the pain space had really had sort of a jaundiced eye for multiple reasons, particularly because there were many failed assets in the past, because there were not good mechanisms of action that had been demonstrated for different assets. There weren't good targets, or there was weak clinical data. And then on top of that, the opioid lawsuits made a lot of large companies that were culpable. Their board of directors told them we're out of pain. And so all of those things were really sort of led to a dearth of innovation, a dearth of investment. Now that there's a peripherally acting agent with an acute pain indication, with an 8 billion market cap readout from just January of this year, I think it's very important because it gives a lot of people the opportunity in the pain space. Things that we and a few others have known for several years, as we've been doing this for several years. And then go to December, Jim, and in December, Vertex also published they put out a press release on their chronic pain phase 2 readout, where the data was not that strong, frankly. In the press release, it was put out to be a success, but the changes were barely 2 points over placebo. So the market didn't quite read it as a success in phase 2 data on chronic lower back pain. And the market cap changed up to 15 billion over 2 days. And that was for chronic pain. So that was also an important market indicator about important these two different aspects of pain are. And then on top of acute and chronic, take neuropathic pain, diabetic neuropathic pain or chemotherapy cisplatin-based neuropathic pain. And that is likely a even larger amount than chronic, maybe even 2 to 2.5x. And so these are tremendous opportunities. Obviously, ultimately, it's for the benefit of the patient so that patients can have something that's efficacious, that will work, and that is safe. And it's important because for the past 40, 50 years, pain spaces had this just the same thing available to it. Now, our approach is different than Vertex's because we work in the midbrain, we work centrally. We know that a radio-labeled SRP-001, which is our asset, crosses the blood-brain barrier. We know where it works in the midbrain, in the pain-sensing area of the brain. They're targeting the periphery. These are important distinctions because likely the future of pain is going to be multimodality. And it's not going to be one single agent. It's going to be sort of like how one treats blood pressure, hypertension, with a few different classes of compounds to have the best control. And so it's sort of like now the perfect storm that's happening towards the pain space that I think will be important. And in the end, the patients will benefit.
Could you tell me a bit more about your lead candidate and how it works?
Yes. So it's a small molecule. It's an NCE, a new chemical entity, novel molecular entity, NME. It's something that we know crosses the blood-brain barrier. We know it gets taken up in the midbrain, in the PAG region, the peripheral gray area. And there it produces something called AM404. And the reason that that's important, Jim, is because AM404 is what gets produced already by something that's being used clinically. So it's a clinically validated mechanism of action. And what we've done is in the library of compounds, and we published this in the 2020 European Journal of Medicinal Chemistry, where we purposely left out SRP-001 because we knew that that was going to be the asset we were going to bring towards all the pre-IND enabling studies and into the clinic, but where we described the library of compounds that this came out of, and we have a few other backup compounds. There we published how it avoids liver toxicity. And the reason is because it has a benzene sulfonamide linker in the middle that avoids production of a toxic metabolite in the liver called a benzoquinoamine called NAPQI. NAPQI's been known since a Journal of Biological Chemistry, JBC publication in 1983 to be the mechanism of action for the dose-limiting toxicity of acetaminophen or Panadol or Tylenol. And so we know it doesn't produce NAPQI. Another mechanism is it doesn't disrupt hepatic tight junctions. And by doing this and producing AM404 in an efficient way, and actually on a molar basis, it produces more AM404, it widens the therapeutic index. And by widening the therapeutic index, we aim to dose higher to treat more moderate severe pain. Again, opioids are up here, the others are down here, and there's a space in between. And that's what we and others, especially the others that are following on Vertex's success with other follow-on sodium inhibitors, are going after, again, to have safer and effective multimodality analgesia, which is probably going to be the future way of treating this.
Could you tell me a little bit about your upcoming trial plans?
Sure. So what we are working to do is to execute a phase 2 in acute pain by the end of second quarter of 2025. And the reason for acute pain is because, as you can imagine, pain is subjective. You know, your pain threshold is going to could be much higher than mine. Mine could be lower, or vice versa. And so it always has to be standardized and made objective. And in order to do that, one of the challenges is always to design the trial so that it reduces variation and variability, so that the endpoints can really be reflective of how good the asset works. And so in order to do that, one of the best models in acute pain is the third molar extraction, the wisdom tooth pull. So for a couple of years, I met a gentleman who's the head of the site, Dr. Todd Bertoch at JBR Cinexcel in Salt Lake City, Utah. And so Todd's extremely experienced, former anesthesiologist, former Air Force guy, and he has actually conducted the phase 2 and phase 3 trials for a lot of the large pharma groups that have taken reformulated, repurposed old drugs. And in fact, was most recently involved with Vertex as well, with their sodium inhibitor. And so he's got a tremendous amount of experience in trial design and execution. So he and I have worked the last several months on the protocol to make sure that it reveals the data as best way as possible so that its true nature can be demonstrated clinically. And so the phase 2 is going to be in acute pain, third molar extraction, with certain primary and multiple secondary endpoints, as well as remeasuring PK to get even more information. We have robust from the phase 1, but just to reconfirm. So it's going to be a very meaty phase 2 data that we expect to actually have a readout well within 12 months of once we start, including data lockout and generating the report to the FDA. And so we're really looking forward to that.
What's the mode of delivery? Is it just a pill?
Oral. So it's oral. It's an oral nanosuspension. And so we've done actually this work with a really excellent group in San Diego called Latitude Pharmaceuticals. The head there is Dr. Andrew Chen, very thoughtful formulation manufacturing chemist. What we've done, actually, I'll just share, is from the oral nanosuspension, we know we can make a tablet, and we also have the steps to make an IV and intravenous nanosuspension as well, and which obviously opened up other pipelines. And the reason that we want to maintain a nanosuspension for these trials is we want to be very scientific about how much we deliver and what the data is and what the PK is and what the endpoints are. And then it's very easy to do if need be any kind of bridging study with a tablet, or if that's even necessary. But there are multiple ways of predicting how the two will work once you have that data. And so, yeah, it's an oral. And then we also have an IV that we've already demonstrated feasibility that it can be done in a clinically formulated way. So as I mentioned, that opens up another pipeline. And then also besides acute pain, we also are targeting neuropathic pain in the phase 2 in the future, as well as obviously after acute pain data from a phase 2 or phase 3 pivotals in chronic pain, which is an even larger market. And so all these things are all intertwined. The focus now is on executing this phase 2 in acute pain.
Do you have anything else in your pipeline other than that?
So we have the backup compounds from the library. Obviously, our focus has been on for consistency, and because of all of the basis from which this is based, all of the molecular mechanisms of action, deciphering that we've done has been on SRP-001. So obviously, as you know, you could change one atom group and then it could behave differently. So, although we have a library of compounds, which is fantastic, and we have actually characterized a couple of them preclinically, all of the molecular mechanisms of action and the preclinical and then the phase 1 data have been based on SRP-001. And we're using that to develop these different pipelines simultaneously.
What do you think the next steps are for, in general, addressing the opioid crisis, and also what you're going to be doing in that regard?
Well, there are multiple things happening. So I was fortunate last year to receive the NIH HEAL directors. The NIH has a program called Help into an Addiction Long Term, which is a trans-agency effort to address the opioid epidemic. And it's in addition to funding, there's also a lot of programmatic things that are done that affect the implementation of clinical protocols and the implementation of information and so on and so forth. And for all of this, I was very fortunate that the NIH HEAL director awarded me one of the 5 Trailblazer Awards for 2024. The reason that I mentioned that is that the NIH has been very active in this space. And then from a regulatory point of view, the US FDA agency granted SRP-001 Fast Track designation a year ago for an acute pain, particularly because it's a novel non-opioid. Looking at all of the phase 1 data, as well as all the mechanistic data, is what we have to show them. So all these things are married because they are happening at the same time. And then it's sort of a perfect storm because then the US has passed some very favorable legislation, particularly the No Pain Act, which basically says for people over the age of 65, Medicare eligible people, just because there's a new entity in the near future or in the future that's going to be more costly, opioids and other medicines that are branded generics are dirt cheap. There has to be the differential in cost has to be basically taken away. So it's a financial incentive to develop novel non-opioids because the reimbursement playing field will be leveled. So all these things are happening at the same time that I think will be important to getting out of this epidemic.
Do you have a timeline for anticipated results from this? Or?
So the phase 2 data is actually quite efficient. We're going to enroll 30 to 40 subjects a month, and it's a 195 subject trial. So as you can imagine, with the getting it started end of second quarter is our aim as we finish the race to getting the data lockout and writing it to the FDA and submitting it. We're hoping well under 12 months conservatively.
I guess it helps that you're doing this study on wisdom teeth and not on something a little more infrequent, I guess.
Right. That's absolutely
Yeah.
right. It's one of the challenges for neuropathic pain. There are different models. We don't have to get into this in details, but, of course, you can do diabetic neuropathic pain. Diabetes is such a broad disease, and there's so many comorbidities and covariables that could really sort of influence the endpoints. And so one of the best ways is something called single axonal neuropathy, which is very rare to the point you're raising. So those are very slow enrolling trials. The challenge is if you use diabetes or even chemocisplatin, again, those subjects have all kinds of cofactors, comorbidities, if you will, that as covariables can really affect the endpoints. And so really you have to balance the two things. And that's why the phase 2 design and the third motor attraction is the way to go for what we're doing.
And what about the difference between short-term usage and long-term? Because obviously with a wisdom tooth extraction, after a few days, it's going to be fine anyway. But then what about for something like chronic
Yep.
pain that's constant?
Right. That's such a large market because that's such a large need, precisely from what you're saying. And that's where, again, one has, I think, rely on data. We demonstrate efficacy. We actually have a multiple ascending dose data as well from our phase 1 trial. So we know that we can dose multiple over several days. And that's when that phase 2 data will inform the construction of a pivotal phase 3 in chronic pain as well to open up that pipeline. The good news is I think having strong safety data with good PK data is where that informs the confidence in the chronic use to which you're raising. The good news is I think there's like as I mentioned, a perfect storm of positivity, which wasn't the case maybe 2-3 years ago, even though we were already working on this even 4 or 5 years ago, because we realized first and foremost the large clinical and societal need. And so all of this I think is very positive, and we expect a lot of continuing good activity in the pain space that hasn't happened for some time.
Well, I guess the more pain treatments there are that avoid addiction, the better. And it seems like South Rampart Pharma is definitely at the forefront of making a difference on that front. That's it for this week's interviews. I did 2 more for the podcast yesterday. I have 2 more later today and a few next week as well. So it looks like we have lots of audio editing to do in the next little while. I haven't decided quite which ones I'm going to air next week. I'll apply that age-old scientific method of selecting at random. On that note, do get in touch. I look forward to hearing from you and your suggestions, and to bringing you interviews with all the top biotech companies from around the world. So, until next time, wherever in the world you are, I hope you have a great week ahead. Thanks for listening, and I hope you'll join us again next week for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.